This Week in Parasitism is brought to you by the American Society for Microbiology at microbeworld.org/TWiP. This Week in Parasitism, episode number 72, recorded on May 22nd, 2014. Hey everybody, I'm Vincent Racaniello, and with me is a pensive Dickson Despommier.
Well, sometimes I get pensive. Good morning, Vincent.
Morning. It is morning. It's 11 a.m.
It's truly morning.
Whoa, so it's a soupy day again.
It's a soupy day. What is with this weather? I mean, this looks like we're living in San Francisco.
Yeah, but there it burns off by noon, right?
This is true.
Here
it just sits around and around and around.
We do need rain, right?
Oh, yeah. Well, speaking of California, they could use a lot of rain.
Oh, they're in big trouble.
They're in huge.
We talked about that in urban agriculture.
Yeah, we did. In
the last episode, we had the one we just published.
Yep.
There's a map of where most of our vegetables come
That's
from.
Right.
It's mostly
That's
from
right.
California.
It is.
And then they have a map of the drought conditions in Cali. It's a mess.
It's a mess. And Texas is not doing
But this
so
is
great
This
either.
Week in Parasitism.
That's right. But you know what? Some of this relates to each other because half of the world uses human feces as fertilizer, and that's a terrific way of transferring parasites from one host to another, especially the ones that are not vector-borne.
As you know, also, as we talked about not too many episodes ago, as climate change progresses, the parasites are moving away from the equator.
They're taking advantage of this opportunity.
Someday we'll have
that we're giving them
chikungunya and yellow fever and dengue here in New York City.
Well, we've already got it in the Caribbean, and people are complaining that they've never had this before, so chikungunya is alive and well, and they're going to confuse it for one other virus infection, and that's dengue, because they've already got that.
At the ASM meeting that I just
How did that
attended
go?
in Boston.
Excellent. That go? Great.
It was a talk called, it was actually an interview. Stan Malloy interviews people for what's called ASM Live.
Right.
You can actually find it at microbeworld.org/ASM Live. It's really they're all recorded in there. And one was with a fellow from the CDC in I think they have a branch in Arizona, right?
They have a branch in a lot of places, that's for sure. The ones I'm most familiar with are in Atlanta and in Fort Collins, Colorado.
Lyle Peterson, CDC, Fort Collins, Colorado. That's right.
That's the Vector
Fort
Lab.
Collins.
That's the Vector Lab.
So he said he's not worried about MERS. He's more worried about dengue and chikungunya.
Yeah, sure, that's right.
And even yellow fever coming back.
Yellow fever, because the same mosquito carries all of those. That's an Aedes aegypti mosquito.
Right. Which are here, right?
They are.
What other mosquitoes do we have here?
How many?
No, not how many. We have Culex species, right?
Oh, you've got culicine and anophelines all over the place.
Anophelines.
Sure.
Aedes aegypti.
Absolutely.
What else? What other Aedes?
Aedes albopictus.
Albopictus, the
is
Asian tiger.
Asian tiger mosquito. That's right. Lots of others Aedes too. Aedes vexans, which is a daytime biter, and that's why it's called vexans, because it tends to bite you at times when you don't expect it. Fortunately for us, it's not been recognized as a major vector for any of the viral diseases that we're concerned about. But, who knows? Viruses can jump from one transport host to another, and probably from one vector species to another, as the West Nile virus did, for instance, when it first came to this country.
All right, well, let's get to eukaryotic parasites.
Let's get some bigger stuff going.
Before we do the paper today, I want to do some follow-up.
Sure.
Or what we like to call FU.
Okay.
But people think we're childish when we do that on TWiP, so maybe I'm going to try it here on TWiP.
Right.
Knowing that we're talking about follow-up.
Right.
That we're being a little jocular.
Let's do that then, shall we?
Can we be jocular?
We can be jocular.
We are professionals, but you have to loosen up. Robin writes. Plethora. So these follow-ups all have to do with the previous episode.
Got it. Plethora. Plethora or plethora?
Anyway, one of us used plethora. So Robin writes, 1540 is a medical word for excess of body fluid. from late Latin plethora or plethora, from Greek plethor, fullness, from plethine, be full, see pleo, figurative meaning, too muchness, over fullness is in any respect. First recorded in 1700.
Wow.
Interesting.
Yeah, that's very interesting. So we have some etymologists out there as well.
Robin is an etymologist.
He's an entomologist and an etymologist.
Then he says, are birds infected? No. So I asked you, are birds infected?
With?
The last parasite, which was Strongyloides.
Oh, right, sure.
Happy trails to you.
Yeah, that's right.
And you said, no, only warm-blooded animals. And Robin says warm-blooded animals includes birds.
No, of course he's right. Of course he's right. I meant mammals. Only mammals. So Strongyloides only infects mammals.
Right. Then if that's what you meant, then why did you say it, Dickson?
Well, I will try to be more precise in my language from now on.
I'm
willing to say I don't know also.
When you're imprecise, well, that's something you do know.
Yeah.
When you're imprecise, people call you out.
You know what?
In fact, we have a few more of that here.
Fair enough. More calling outs?
For example, Rachel writes, Dear TWiP team, I'm skipping an email just in case you want. In listening to TWiP episode 71, whilst gently tending my cell culture garden, I noted an inaccuracy in your classification/description of birds. In the context of the host range of Strongyloides, Vincent asks, are birds infected? To which Dickson replies, no, warm-blooded animals, implying that birds are not warm-blooded animals. Oh. This is a mistake that I have noted being made in previous episodes of the Twix family of podcasts as well. Most birds are indeed warm-blooded animals, more precisely homeothermic endotherms, as are most mammals. A one-time point in need of clarification in the same part of the conversation, which seemed to be about Strongyloides in general, not S. stercoralis specifically, is that while it is true that S. stercoralis only seems to infect mammals, there is at least one species which does infect birds, S. avium.
Right. I can add to this conversation, too, because when I was a student at the Michigan Biological Station in Pellston, Michigan, there was a guy there studying swifts, or swallows. I think swallows in this case. Purple martins. No, I'm sorry. It was actually purple martins. So purple martins is either a swift or a swallow, but it's related to those types of birds. They're very fast, and they're insect eaters for the most part. And when they're born, they are poikilothermic.
Poikilothermic, which means what?
Which means that their body temperature fluctuates with the outside temperature. And then as they progress into their life as a small purple martin and eventually into an adult purple martin, they convert to warm-blooded activity as their needs are different, obviously. So I learned that term there, and I thought I would never have the opportunity to use it, but here we are. And I'm fully aware that birds are warm-blooded, and I'm also fully aware that mammals are warm-blooded. And so I didn't mean to confuse the audience by saying that only warm-blooded animals can get Strongyloides stercoralis, but rather mammals are the only group that can harbor Strongyloides stercoralis.
Now you've learned too. Well, you already knew that, but now
well, that's fine. No, listen. These are small corrections of facts that are totally worth the discussion.
Rachel is a DVM PhD who is at the University of Iowa.
Great.
All right, our last FU today is from David, who writes, Dear guys, question, who is Spencer Wells? Arose in final minutes of TWiP that I just heard, near mention of trogocytosis and a couple of enjoyed PBS series. So remember last time I said, who's Spencer Wells?
Right.
You didn't know either.
No, I didn't.
Spencer Wells also had a PBS, I think, bit of his production. Must be something 10 years ago now, but charmed me so that it seems fresher than that. I've always wished to see him back with more. I wish I knew the title. He is a geneticist and I guess was on the cutting edge of reconstructing the origins of human dispersal. He clarified the first spread of sapiens along the South Asian corridor, ultimately to Australia. Your context was tropical species which went where it didn't need clothes. His bearing seemed a little American aristocratic, but not in an offensive way. I thought his discussion was especially smart, and I felt exceptionally well informed by the show. Is it conceivable that it was a bit too far above intellectual middlebrow to satisfy the viewing demographic? I don't know and shouldn't even speculate, and must add that I too have enjoyed the other PBS productions you guys named. Besides, Human Origins has always been a favorite hobby horse of mine. Anyway, the genetics-based reconstruction of human population origins has moved by leaps and bounds since, as you know, yet I haven't encountered any but snippets of detail in public productions. I wish Spencer would come back with a new show on the subject. That's who I think he is. Fondly, Dave. P.S. So what is trogocytosis? Now, we'll do that paper another time. How about next time?
Sure.
I have to remember. I would like to do a podcast on human origins.
That would be fabulous.
With Spencer.
Right.
Now, here's the thing. Spencer had a PBS production or whatever, national, right?
Yep.
And if you get too complex, they don't want you. They think that you have to dumb it down to get the most number of listeners. And that may be true, but, here on the TWiP podcasts, we get lots of people with long shows, although we don't have millions of people.
Oh, that's right.
And I think at our level, we're not going to get millions of people.
No.
Which is unfortunate because it would help to support our endeavors.
Do you know which show did, though, that it's about parasites? It's called Monsters Inside Me, which was a Discovery Channel show, which I was the science advisor for.
So were those like an hour episodes?
Yeah, they were an hour of case histories, basically. There were several people who got infected with certain parasites, and we can put a link to it in the show notes.
Who did the show?
It's a good question.
Well, I remember we've talked about it before because of
the host of the show escapes me. His name escapes me. He did a good job.
Why can't you do a show like that?
I could.
Why does it have to be somebody who never did a parasite
I think
experiment?
casting for these shows they're based on.
You're kind of ugly. That's
know.
the problem. So am I.
Yeah, well, that has nothing to do with the choice of show host.
No,
Trust
of course
me.
it does.
No, that's not true at all. No, I wouldn't agree with that at all.
Well, the news readers are all attracted.
I don't know why they chose the guy they did, because he did a good job and he explained everything right. And when they had the case histories with misinformation, it was my job to look up the facts remember them and then let them know. And they got quite a bit of stuff wrong to begin with, which I felt I helped the show with. So they were into their third year, and they had millions of viewers, by the way, millions. So that's basically, you have to capture their imagination somehow. And the Monsters Inside Me title was enough to get people to look.
Of course, it's a great title.
Right.
Well, I would like to have a broader audience only because, first, I think that's our goal, to teach people about science. And it would help us to support the shows because we're now reaching the point with 4 podcasts and a 5th soon to come online.
That's right.
And I have at least 2 more ideas.
Okay.
I can't do it all myself anymore. No. I need some help, and to do that, we need money, and so.
Well, this is not a pledge week for us, but
no, we're not asking anyone to give money.
No, we're not.
But anyway,
right.
All right, we have a paper today, which is very cool.
It is.
It's a Pearl.
I have a paper version of it right over here.
It's called a Pearl.
The Pearl.
It's published by PLOS Pathogens. They have this series called Pearls.
Okay.
Which, and to take from their description. And then, by the way, PLOS Pathogens, all the PLOS journals are open access, so you can go and look at them anytime, which is great.
Right.
I must say the authors pay for that privilege, though.
Sure.
We pay thousands of dollars to publish an article.
Yeah. What does PLOS stand for?
Public Library of Science.
See that? Okay. So it is open to the public.
It's open access, but the authors pay to publish their papers.
Right.
And that's their model.
Right. Well, we pay to publish our papers in other journals.
I think this is perverse, to be honest.
We do.
This is perverse
to
publish scientific findings, which are then picked up by companies or whatever to make human life better. We have to pay to publish that?
I hear you.
I understand it's the model.
It is the model.
But it's perverted.
Right. Well, usually the journal is produced by some private association.
Well, PLOS is not. PLOS is public
library.
And it's online. It's online. It's a nonprofit.
So you're paying for the maintenance of the website.
Yeah, but I think they need to work on this because they have hundreds of employees.
Right.
They have all this front matter, including pearls.
Right.
But I just would. There's a great thing called bioarchive where you just upload your paper.
Got it.
And it sits there, minimal cost.
Got it.
I don't need all the fancy layout and a staff and.
Yeah.
This is crazy.
I'm sure you're going to get a lot of letters and emails.
No,
on this
I'm not. I'm not.
No, maybe supporting you.
Oh, okay. Well, they can not support me. It's fine, but I've published in science for 32 years.
Yeah, me too.
And I've spent tens of thousands of dollars. Now, someone's going to say, well, you take that money from your grant. I understand. So you build it into the cost of doing research to publish. So you're supporting the journals, right?
Right.
So that's another way to look at it, and maybe if I looked at it that way, I would calm down. I just think it's perverted.
No, I don't think you'll calm down.
You have to publish it. Every journal has a different price. PLOS is very expensive because that's the only way they support. But there have to be other ways to do this, which I think is reducing the cost. I don't need no fancy layout.
Yeah.
I don't need any front matter reviews or perspectives. I don't need any of that.
I got it.
I just want the paper.
I got it.
You want me to shut up?
Nope. You have anything more to say?
I'm sorry. Well, I'll edit this out if you want.
No, fine.
The paper is called Wormholes in Host Defense, How Helminths Manipulate Host Tissues to Survive and Reproduce.
Exactly.
And these pearls are very short summaries, which are meant to be written in an easily digestible language. So not a lot of science detail.
No.
We're going to go through this, and we're going to supply some science detail.
We will try whenever we can to do that.
Yeah.
But I think we should give some background first.
Yeah, of course. I was going to ask you, what's a helminth?
Well, true. As a virologist, that's a valid question. So helminths are worms, obviously. All right. So that's a general name.
And that's the end of our show.
And well, are there any questions? That's right.
Helminths are worms, which we have talked about.
Of course we have.
Give us an example. Strongyloides.
3 kinds of
last
worms.
time. Strongyloides.
Yeah, that was a worm. It was a round worm.
3 kinds: round,
flat,
and
segmented.
triangular.
Flat. No, not triangular. Sorry.
Segmented flat.
Rectangular. No, that's not
round,
true.
Flat, and segmented flat.
That's right.
Strongyloides is
a roundworm, nematode.
Nematodes. Why nematode? Where does that come from? Captain Nemo?
I'm not going to venture a guess here. I used to know the answer to that question, but I'm sure we could look that up because it's a Greek derivative.
Well, Robin will probably tell us.
Yeah, Robin would. Oh, maybe we should have him on as a co-host.
Well, he could be on the line right now and supply all the...
Nema, I think, relates to roundness. Nematode. The word toad is a derivative from all of the worm groups. You have cestodes, trematodes, and nematodes. It's just regular tree toads. No, that's not right. That's a frog. So we have these categories.
We talked about
of helminths.
Nematode last time. We have talked about all the nematodes.
We did. So classification is, started out in the old days when they could see these worms because a lot of these things are visible to the naked eye, like Ascaris, for instance, which is a roundworm, and tapeworms are easy to see, so it's easy to see those too, right? And the
is Racaniello a worm?
Of course.
Which one? What kind?
A nematode or a nematode. Either way is pronounced correctly. Okay. So in all those early days, Trichinella was one that was discovered by microscopy. All right, because it's too small to see with the naked eye, basically. You don't know what you're looking at, and so you have to use a hand lens or a microscope to see it. And we discussed the discovery of Trichinella on this show a long time ago. So, but the point is that nematodes, cestodes, and trematodes are, for the most part, well, I was just about to say something that I know isn't true. So if you go to the nematodes first, most nematode species are free-living. That is to say, they're not parasitic. They live in soil. They live in a variety of environmental habitats that have selected for their lives with regards to the temperature, humidities, and other organisms that happen to be there, the essential niche, so to speak. So Rhabditis elegans, which is the genetic organism with respect to...
It's not a genetic organism.
Well, an organism that has been selected for studies on genetics because it's easy to manipulate.
I'm going to keep you right.
Okay. Listen, you should keep me right. And don't keep me left.
The thing about you is you have always a good sense of humor, no matter what I do to you.
It doesn't... Well, that's in public. You should see what I do in private. No, that's not true.
You never yell at me. You never get mad.
Why would I do that?
As opposed to me. I get kind of cranky, right?
Vincent, you're absolutely fine. No, I think we have a good relationship here. So Rhabditis elegans is the organism of choice at a certain level of life if you want to understand that whole group. So you're understanding all nematodes when you work on Rhabditis elegans in terms of the biology, okay? But it's a free-living worm. It's not a parasite. It lives in soil. It eats bacteria. That's what it does for a living. And it reproduces like crazy. So there's tons of them out there. And there's a lot of other examples that are related to Rhabditis elegans that are not parasitic. Now, the last show that we did was on Strongyloides, right?
Right, of course.
And Strongyloides is confused because it wants to be like Caenorhabditis in part of its life cycle, and in another part of its life cycle, it's totally parasitic. Okay, so there's a small fraction of the myriad species of nematodes, and I challenge anyone to submit us with the exact number of species of nematodes, because I don't think we know. There are just so many of them out there.
Well, I think that's true for everything, right?
We probably know most of the nematode species of mammals. Mm-hmm. We know most of the nematode species of birds. Most. I say most because not all.
Which are not mammals, by the way.
Which are not mammals, but they're still warm-blooded beyond their poikilothermic stage of development. So, and then we know a lot less about the nematodes of fish and ocean mammals. We know, but we've had a show on that too when we discussed the dangers of eating unprocessed sushi, as you'll recall. So, so that's nematodes, all right? And that just refreshes our audience's recollection of what we've already discussed. So then we have cestodes, which are tapeworms.
Right. Like
now,
Trichinella.
Unlike the nematodes...
No, sorry, Trichinella is not a cestode, right?
Trichinella is a nematode. It's a round worm.
Got it. The flat ones are cestodes. Cestodes are not segmented?
Segmented flatworms
are cestodes.
Cestodes.
What are the non-segmented called?
Those are the trematodes.
Trematodes. I should know this, but I'm just asking you.
You don't know this. Why should you know this? That's why I'm here. I can be your guru at this level.
I'm asking you as a mechanism of teaching.
And we want all of our listeners to know that we're not sitting here reading books, by the way. We're just recollecting this, okay? So this is part of my background as a scientist. I had to learn this early
that's why
on.
you are good on this show, because you have all this knowledge of years.
Yeah, well,
and you can just turn on the faucet.
Yeah, turn on the faucet, and sometimes the wrong fluid comes out, and we have to get corrected.
But you didn't work on birds.
No, I didn't work on birds. So
do you think that we should know everything?
I hope not.
Yeah.
No, that's why there's such a plethora of knowledge, and people with that knowledge form the basis for our existence.
Cestodes.
Every cestode is a parasite.
That we know of.
Right. We don't know of any, none.
Well, there may be some out there that we haven't discovered.
I don't think so, Vincent. Every time we've looked, we've discovered only parasitic versions of cestodes.
I bet near the thermo vents at the bottom of the ocean, there are non-parasitic cestodes.
No, because they all live in the gut tracts of animals.
Well, the worms down there have gut tracts.
I beg your pardon? You mean...
Not the worms, those big tubular
well, things. Those are tube worms. Those are interesting organisms, right?
Very... Very interesting, but we digress.
So the point is that as far as we know, let's put it this way, as far as our knowledge goes, all cestodes are parasites.
All right.
So that means that they have to adjust to a host somehow. In that little fraction of nematodes, they have to adjust to their host too. Or one of two possibilities. One is that they can manipulate the environment of the host to suit their needs, or they can sort of just fall right into the niche that they happen to find themselves in.
Okay.
Cestodes, for the most part, as adults, are well adapted for the gut tract of the host that they live in. Okay? And in fact, their outer surfaces often resemble, in microstructure, the surface of the gut of the host. They have microvilli, they have an absorption surface, because cestodes don't have gut tracts. All right. Now, trematodes, on the other hand, okay? Most trematodes are parasitic, but there are some that are not. And planaria is one of those organisms that's a flatworm.
Lovely.
That's related.
It's another model organism, right?
Yeah, that's another model organism. They use it a lot for learning experiments and things like this because it has a developed nervous system, okay? And they do a lot of studies on that. But all of the schistosomes, for instance, those are all parasites. And all the monogeneic and digeneic trematodes, those are all parasites. So for the majority of the life forms in Trematoda, they're parasitic. All right, so we've got these three groups, and they're constantly trying to infect hosts, constantly, with their infectious stages. So the question arises immediately, how do they succeed? Remember, we had these two groups of parasites, the long-lived ones and the short-lived ones? Mm-hmm. And I'm really not interested too much.
It was like episode 2.
Yeah, exactly.
Wow.
So I'm not terribly interested in the short-lived parasites because they either succeed or they don't, and they cause disease or they don't, and then they're eliminated or they eliminate you. And in doing so, they eliminate themselves. So that's not a very strategic life cycle for a parasite to adapt to. Okay, so the point is that we're really interested in the long-term survivors, and this article addresses those long-term survivors. How do they do it? How do they manage to survive in the face of a highly developed immune system that's both innate and activated by the presence of antigens, right? Adaptive.
Right.
So you have these two arms of the immune system, these two basic arms, and they fight off maybe 99.9% of all the invaders because they're not adapted to fitting in.
Right.
Every now and then, though, you've got a mutant from another group that all of a sudden has an advantage. And one of those adaptive mechanisms is controlling the immune system of the host.
Yes, modulating it.
Or
right.
hiding from the immune system.
Yeah.
And we have chronicled a lot of these strategies in our descriptions of these parasitic infections. And so we've had a lot of fun talking about the mechanisms involved and everything else. But here's a very, very condensed summary of the examples that they have pointed out to the audiences that they would like to reach. Okay, these are two scientists at Stanford University, and
Deborah Boyett,
right,
and Michael Shih.
Is that?
I don't know how to say it.
Michael or Michelle?
Oh, it's Michael.
Michael. Right.
HSIEH, both at Stanford.
Absolutely right. And
5 themes of helminth manipulation.
Exactly, Vincent. So you've read the article, and I read the article. Yeah. What did you glean from this?
Well, let's.
Shall we go through them?
I want to go through it.
Let's do it.
But I want to start with their first sentence, which is over 1 billion people and innumerable other animals are infected with one or more helminths.
A billion.
1/7 of the world's population. Do you think that's accurate?
No, I don't at all. I think it's underestimating tremendously. Why? Because I'll tell you why. Because almost everybody in the world encounters one nematode parasite in their lives. As children.
Have I?
I think you have.
Which one?
I'm sure if you just think a little bit, you can recall that one.
I don't like to think. It hurts.
Because you've got children.
I do.
They grew up in your house. I'm sure that they had this parasite. And I'm sure that you and your wife sort of said, oh, my God, they've got pinworm.
Oh, pinworm. Yes, of course.
Pinworm. That's a parasite.
I don't remember.
It's a it's a passing phase of our lives, basically, because as adults, most of us are not susceptible to that parasitic infection. We talked about that. We did. So that's 7.15 billion people that have at least had one nematode parasite, right? So everybody on this planet, unless otherwise proven by some,
what is it that you said a long time ago? When there's life, there's parasites?
Yeah, successful systems attract parasites. And we are a highly successful system, a biological system on this planet. And we have attracted our fair share of parasites.
Whenever parasitism is possible, it will happen.
Well, that's right. So by selection, we're it.
It's a weird character of life, isn't it?
Yes.
Being parasites. And you see it all the way up to us. We're parasites.
You do.
My son asked me this morning, my
Yes.
younger son, he said, Dad, what's a parasite?
Oh, really?
I said, you're a parasite.
Oh, he said, people who live in Paris.
No, I didn't say that. Viruses or anything that takes from another organism hurts it, right?
Well, or that takes advantage of it.
Takes advantage, but it
That's
does
correct.
inflict some...
Often
No,
it
because
does.
of course the parasitic infections we've talked about that last the life of the host, and there's no problem.
I know. So
this idea of a parasite having to hurt is maybe not.
well, yeah, those are leaning towards endosymbionts.
Yeah, mutualisms, right? Well,
so
we give them a place
pinworm,
to
for
live.
instance, is the adult worm itself is thought to be benign.
Yeah,
it doesn't cause any harm to the host whatsoever. But
we may help. These may help us. We've talked about ways that they can help us by suppressing the immune response,
or by stimulating both arms of the acquired immunity. TH1 and TH2 responses are both stimulated. We have one parasite here, at least, that does that as a way of ensuring its own survival, I think, or at least it does. Whether it does it on purpose or not, that's other.
So they have 5.
They do.
Themes of manipulation. The first one is disruption of epithelial barriers.
Right.
So
how do you gain entrance?
How do you get through? Right, because we have the skin, we have mucosal membranes that are barriers.
So what are the routes of infection for parasites then?
Well, skin is a big one, right?
It's a big one, but it's not
skin,
the
lung.
most common.
So they say skin during Cercariae invasion.
Indeed.
Cercariae.
From schistosomes.
Only schistosomes. Yes, schistosomes.
Sure.
The lung during transit, they say, of schistosomes.
That's true.
The intestine and bladder when they come out, right?
Yes.
So these are all multiple transits of the epithelium.
The eggs themselves have to find their way out of the body, and they're trapped inside the body because these worms live in your circulation.
So, for example, they summarize some studies where the Cercariae are believed to have proteases that degrade the elastin and collagen in your skin to allow them to get in.
This is right.
It makes perfect sense, right?
It does, but there's another group here that I hope they're going to
Viruses don't do that. Viruses
do
to
not
get through
do
the
that.
skin, they either have to be delivered by a mosquito or a scratch or a needle.
Or in a cell that's already in that suspension of what they're injecting, right? So,
I'm sorry, what were you going to say?
No, though, there's another group of worms here that also have to penetrate the skin, and Strongyloides was one of those from last time.
That's right.
But hookworm, too.
Now, wait, before you go to Strongyloides,
sure.
Schistosomes, they also talk about how the eggs might get out of the bladder.
Right.
There's evidence that the eggs in the ova cause decreased transcriptions of genes in the lining of the bladder that make the cells loosen up, basically. Because, there's a tight barrier there composed of tight junctions.
It's transitional.
And so they shut down genes that are needed for that, so it kind of loosens up so the eggs can get out. Isn't that brilliant? That's evolution for you.
It is. And there is something inside the egg, all right, that's doing all of this. And it's a larva that's already developed. And the larva is called the myricydium. And this is the stage that when the egg does finally get out, will hatch in water, fresh water only, and then swim and follow the chemical trails that snails leave. And they're obliged to find their snail hosts in order to continue the life cycle. Right. So this larval stage, though, has multifunctional abilities. It can not only alter the genetic expression of the transitional epithelium of the bladder and probably in the large intestine as well, because they collect in packets from Schistosoma mansoni and japonicum there. So hematobium is around the bladder, but the other two species are elsewhere.
Okay.
So, but then when they get out, they have to now survive in a freshwater situation. They're free living.
Mm-hmm.
They have no growth metabolism, but they have a maintenance metabolism, obviously. And they have a specified length of time in which they must find a snail host, otherwise they're going to die. But once they do find the snail host, they have chemoreceptors that allow them, that guide them to the host. That's a homing device, right? Like a heat-seeking missile. And then once they get in contact with the snail, they now have another job, and that is to get past their epithelium. So it's not just our epithelium that this parasite has to traverse, but it's also snail epithelium. So it makes you wonder, who were the first parasites of this parasite? I mean, who were the first hosts of this parasite? Was it snails first, and then later on they evolved to infect mammals? Right. Or, because mammals are late in life, basically snails have been around for a long, long, long time. Pre-Cretaceous era.
So they may have been the first then.
Yeah.
Right.
It could have been. Did I say that right? Cretaceous? I meant Carboniferous. But that's okay. You see, this is where I'm skating on such thin ice that I'll never be able to actually stay afloat.
You have a broad
right. So for millions of years, hundreds of millions of years of evolution, prior to the time that mammals arose, this parasite may have been evolving towards a life in snails only. Right? And then when mammals came along, or some other opportunistic organism like reptiles, for instance, or dinosaurs, which were not reptiles, by the way.
I know.
Then, I know you know. Then these organisms might have attempted the passage and succeeded, and they may have had schistosomes as well.
Now, they say, in contrast to skin invasion, many helminths have to go across host epithelial barriers after being ingested, like the whipworm.
Let us talk about
Trichuris trichiura, which I spent a long time on that episode pronouncing.
Right.
practicing.
Right.
And finally, Trichiura.
That's right.
Trichurus. Anyway,
Sure.
They secrete proteins that alter intestinal epithelial cells.
This is a remarkable adaptation, right? Because the worm itself, when it starts out, is microscopic.
How do you pick up a Trichurus again?
It's from fecal contamination.
You ingest it, right?
You ingest the egg, which has a viable larva developed inside the egg.
Okay.
The eggs are not passed that way, though. Their eggs are passed fertilized but unembryonated.
Mm-hmm.
And then in the soil, along with the feces that they've been deposited on, they develop further. And it takes them about a week in order to go from a fully fertilized egg to a fully embryonated egg. It's about a week embryonation period. And now the eggs they can last a long time in the soil. They can be there for months if the conditions are correct. And then you ingest the egg. And then the egg is permeable. And it allows us signals to go through the egg into the larva that triggers the induction of enzymes, which allows the larva inside to then hatch out of the egg when it's in the right place in the gut tract.
Well, listen to this.
It's pretty bizarre stuff, right?
One of the things they talk about here is that the proteins secreted by the larva establish a pore in the.
It's a pore-forming protein,
so that the larva can stick its head in, or not a head.
Well, no, it is a head, of course.
It can insert itself through.
It does.
Right? It's amazing.
And
this is in the gut tract.
In the gut tract, in the large intestine, and once that pore-forming protein has been secreted, and by the way, there's another parasite called Entamoeba histolytica, which invades the same cells and kills them, but it also secretes a pore-forming protein, but it's not related to the one that Trichurus uses, because Trichurus wants to set up a long-term relationship with its host. Entamoeba histolytica wants to either eat all the cells it can and reproduce and get out of that host pretty fast, or it obviously can remain as a smoldering infection in some instances also. But for the most part, people who encounter Entamoeba histolytica either get killed by it or they cure it within a several-week period of time, whereas Trichurus can last for up to 3 years in a host. 3 years is a long time.
It is.
And it's sitting down, nestled among the Peyer's patches, for gosh sakes. Those are our immune defense systems in the gut tract. Why don't we kick this worm out? Why don't we kick this worm out? And the answer is not obvious, because we haven't discovered all of their escape mechanisms yet. But they're not the only ones that have to traverse the gut tract in order to infect the host.
These are just some examples where we happen to know how.
So the most common of all the worm infections in the world is Ascaris.
Okay.
The most common besides Enterobius. The one that's pathological, these are the big worms that are about the size of a pencil. When you swallow their eggs, okay, just like Trichurus, goes into the small intestine. It receives a signal from the environment that it's in the right place. The worm inside secretes proteases, which allows it to exit the egg. And now it has to traverse the small intestinal epithelium.
Right.
And it has proteases for that purpose as well. So Ascaris is one of those big infections and then it traverses the body and goes to all kinds of places. We've chronicled the life cycle of this particular parasite. It goes to the liver first, and it goes to the lungs. It has to get out of the lungs into the trachea, up to the bronchi. You swallow it again, and now it's not an egg any longer. It's a third stage larva. It molts another time in the small intestine rather, and a fourth time it molts 4 times, and now it's an adult. Takes it 3 months to grow up from a larva that you can't see to an adult the size of your pencil. And at that point, they start producing eggs. Trichurus is related to them in the sense that it takes a long time for the adults to grow up. They're slow growers. It's 90 days by the time you encounter Trichurus before you're going to see eggs in the stool. Okay, so that's a long term. That means this parasite has to find either a niche which is non-immunologic, so that it doesn't get kicked out, or it has to modify the immune response to allow it to survive in the face of, it's like looking down the barrel of a gun, basically. And somehow you have to take the bullets out of the gun. Otherwise, you're going to die.
Nice analogy, Dr. Despommier.
We try to teach this way so that people can envision these things for themselves.
The last example here.
Yep.
May I?
Yeah, of course.
Strongyloides.
Sure.
Perturbs mucosal integrity.
You bet.
It disrupts the balance between death and growth in the small intestine.
Right. Now there's...
Right?
When Trichuris... no, I'm sorry. I'm going to back off here. Trichuris lives in this multicellular niche that it induces for itself in the large intestine for up to 3 years. That's what I just said.
Right, right.
So there are 2 other examples of worms that live in the small intestine, which disrupt epithelium. One of them is Strongyloides, of course. Where do they live? That's the point. What is their niche of the adult Strongyloides? Where does it live? And there haven't been good studies on this, Vincent. There have not been. But the supposition is, because of the histology of the infection, you can tell that it's an intimate contact with the epithelium. It may be living in the epithelium.
Duodenum and upper jejunum.
Correct.
That's what it likes.
Correct.
Those are parts of the small intestine.
They are. But where in that organ?
So here, explain this, can you?
I'll try.
It says, and this disruption of cell death and proliferation balance may facilitate autoinfection in which the Rhabditiform larvae penetrate the mucosa to perpetuate infection. Can you elaborate?
Well, the Filariform larvae are basically the ones that are infectious here.
What does this mean, autoinfection?
Autoinfection. Well, we talked about that with our Strongyloides episode, but
it was more than an hour ago. So
I'll come back. I'll come back to it because in certain circumstances, and they're unusual circumstances for the host, by the way, one of them involves the blockage of the large intestine with a bowel tumor. It slows down the transit time for the feces that's formed above that tumor. And so it allows the worms inside the... remember, Strongyloides gives birth to live larvae. Okay? So the live larvae, they penetrate out of the tissue into the lumen of the small intestine, travel to the large intestine, are included into the fecal mass, and they exit the host. That's their life cycle.
Right.
But what if there's a block? What if you slow down the feces on its way out?
Right.
You know what happens? The worms still develop. And instead of developing to a 3rd stage Filariform infectious larva out of the host, they develop to a 3rd stage Filariform infectious larva in the host. And that's very bad for the host because you can now accumulate more and more adults as the result. Sure. And in addition, the worms migrate throughout the tissues, and when they change to the 4th stage, they get rid of all of the microbes in their gut tract that they accumulated from the Rhabditiform stage to go to the Filariform stage. I know it's confusing for these terms, but it's like looking at viral synthesis. That confuses me somewhat too, because I didn't grow up with that knowledge as a scientist. Okay, so the point is that the Rhabditiform stage of worms that have this kind of life cycle feed. And so what do they feed on? They feed on feces. The Filariform larva is not a feeding stage. It's an infectious stage. And how do you transition from a Rhabditiform to a Filariform worm? You shed your cuticle. There's some development that goes on inside the worm before that happens. And all of the contents of the gut are shed, gotten rid of. Well, that's bacteria. And so people with autoinfectious strongyloidiasis sometimes die from overwhelming bacterial sepsis. And it depends on the species of bacteria. And remember, the human microbiome is huge, right? It's got about 1,500 different species of bacteria in your gut tract. You could die from any number of those being deposited in places throughout your body, like your brain tissue, for instance, or your liver. Right. So that's the details of what they're talking about here. There are situations. Okay, that's one. A bowel blockage is one. But number 2, one of the most common old ways of getting an autoinfection started in a host is to transplant their kidney. They come in for a kidney transplant because they've got diabetes type 1, and they don't know this, but they have a smoldering Strongyloides infection. When you give them cyclosporin A or some other even more powerful immunosuppressive agents, the worms develop faster, which tells you something about the control of the host immune response on the development of the worm. Our immune system holds this worm in check all the way out of our bodies so that it doesn't develop to a third stage larva. The moment you inhibit the immune system, and we're not sure of which part of it we inhibit by these powerful immunosuppressive drugs, the point is you release the hold of the immune system on the development of the worm, and it can now develop to a third stage within the host. And again, you're into this same horrible problem of this autoinfectious cycle. Right?
Got it.
All right. So I think we're these are the pearls, and we're now filling out the entire necklace of information with regards to each one of these things.
Number 2, reinforcement.
Oh, we didn't finish though, because I have to re-mention the fact that, remember, it's not just Trichuris that modifies the epithelium, so the epithelium doesn't die.
Correct.
The Strongyloides modifies the epithelium, and the epithelium does die, but there's a third one, and that's the adults of Trichinella. The adults of Trichinella spiralis can last for about 2 to 5 weeks, depending on the immune...
Well, that's a separate...
Yeah, but I'm talking about the epithelium for the adult worm right now.
Right, okay.
I know there's a separate one for the larva of Trichinella they discuss as well, but the adult worm lives in a similar niche as the Strongyloides adult. And that niche is an intra-multicellular niche. If you measure how long these worms are, and in the case of Trichinella, they're about 3 millimeters long for the adult. That's 3 millimeters long's worth of epithelium that it takes to surround this worm. It lives in the bodies of the epithelium itself. What a remarkable, crazy niche to pick as a nematode infection. It's an intra-multicellular parasite at that point. And I think Strongyloides is as well. And we don't understand those niches very well at all. There has to be much more study with regards to that, because they obtain all their nutrition as the host is obtaining their nutrition. It goes across the host epithelium through the microvilli, and the next thing you know, you're in worm central. You've got this big, gigantic organism living inside you. The Trichinella has some remarkable cuticular pores that go along the entire length of the worm in pairs. 2 pairs are dorsal, 2 pairs are ventral. They're called hypodermal gland cells. We have no clue as to what they do. My suspicion is always, as has been, not only are they osmoregulatory for the worm, I think they can absorb nutrients through these pores. And that's how I think they get their nutrition. But that's just a speculation on my part, okay? And I would love to see somebody do some actual studies on that to show this. And by the way, Judy Appleton and her colleagues up at Cornell University, which is where you graduated from school, I know this, Vincent, she has an in vitro model.
Yeah.
To study the adult worm in a lawn of contiguous epithelium. So she could actually do these studies.
Why don't we go up there and interview her?
I would love to do that.
Drive
She's an old friend. And
a couple of cameras and a mic.
It would be great.
You know, she could show us
stuff. It could be old home week that you got. You know, I would love to do that.
It's not very far. We could drive up.
We could do that.
All right, let's do it.
Sure.
We need a film crew.
We need a film crew. Right. We're working on it.
Number 2.
Number 2.
Reinforcement of epithelial barriers. So in contrast to disrupting them, there are apparently some helminths that reinforce them, and that makes the host live.
Correct.
So that it's good for the parasite. So they say Schistosoma japonicum, the eggs have the ability to increase the impermeability of the colon, the epithelial barrier of the colon,
right?
And that decreases the ability of bacteria to get in and kill the host, which you were talking about a little bit about before, right?
Yeah, when you create breaks in this defense system, and one of them is just a barrier.
Bacteria come in, absolutely.
Like crazy, right? I mean, they do anyway, because that's why the liver exists.
Yeah, sure.
With the P450 enzymes to take care of all these horrible products that are coming in from the gut tract that bacteria are producing. We can depressurize all of that stuff, right, by detoxifying.
So there's the way the epithelium, mucosal epithelium is constructed, there are tight junctions, right?
Of course.
So you
have to have tight junctions.
And there's a protein that's part of them called occludin. Nice name, right?
Yes.
And it's thought that one of the schistosome proteins triggers an increase in the amount of occludin to make it to reinforce the barrier. Isn't that
fantastic.
Cool?
Yeah, it's very interesting.
Very nice.
Very interesting.
Also, they say that S. hematobium,
yes,
the other schistosome,
right?
The presence of eggs in the bladder triggers proliferation of the cells lining the bladder.
That's interesting.
It's kind of a resealing. So the ova caused some damage, right? And this reseals it. Fix it.
So
It's great. You go in, you make a hole to get in, then you patch it up.
Yeah, well, actually, they're coming the other way, though. They're not going into the bladder. They're going out of the bladder.
Sorry. My apologies.
Not at all.
I'm just so excited that I got the directionality wrong.
That's okay. But there's a consequence for increasing the proliferation of they're called transitional squamous cell epithelium. There's a downside to this.
What's that?
Vincent. Every now and then, one of them makes a mistake.
Yeah.
And what do you get? You get bladder cancer.
I was going to say you get a tumor probably. Yeah.
You do.
When you make
so
a
lot of
mistake,
people from schistosoma hematobium die.
Because there's a lot of proliferation. That's the key.
Exactly right.
When you get uncontrolled cellular proliferation, boom, tumor.
That's right. That's right. Because every now and then,
as cells divide uncontrollably, they accumulate mutations.
See that?
And you just have the right 10 or 12 in the right genes and you become a tumor.
You deregulate that phosphorylation cascade to division, and you're off to the races.
They say here, besides nematodes, which schistosomes are, right?
Yes, of course.
Okay.
You bet.
This kind of, this is called hyperplasia.
Yes.
Making the tissue proliferate. Has been found in trichinella.
Right.
Nipostrongylus brasiliensis, which I don't think we've ever mentioned.
No, but it's a rat parasite that's used as a model for hookworm.
Oh. And here's another one. Heligosmoides polygyrus. Wow.
Helix. Right. It's a tongue twister. Helixmomoides.
Helixmomoides.
Polygyrus. That's another intestinal dwelling nematode infection that's often used as a partial model for hookworm also.
Okay.
Right. H. polygyrus is the way they always pronounce it. Forget about the first word.
Now, Nipostrongylus, Nippo probably refers to Japan, which is where it was first discovered. Nippon, right?
Might be.
And then Strongylus is part of
There's a way to look all this up, because you go to the parasite websites and you can find out where these names all came from.
Trichinella.org.
Well, I know where that name came from. It was originally called trichina, because trichina is the Greek word for tiny hair. But as it turned out, that word had already been used for another organism, so they had to name it trichinella, even tinier hair. That was Richard Owen did that one.
Number 3.
Yes.
Alteration of lymphoid tissue.
Ah, very important, Vincent. Very important.
S. Mansoni are believed to remodel Peyer's patches in the gut. And that way they're better for their eggs to be put in. They mess with the vasculature, which is the circulatory things, right?
They're not happy. These are not happy with their new homes.
No,
they have to remodel everything.
Yeah, they come in and they get
what is wrong with these people?
Old house. What was that guy's name?
That's right, Norm
Abrams comes in. No, actually, he was the assistant, but he did his own show, the Yankee Workshop.
Yep.
Who was the original crook on This Old House? Remember?
Oh dear, hard to remember. But no, wait a minute.
He was a crook.
We've got the. Oh, I know who it was. It was. He eventually ended up working for Sears. You're going to find this, I'm sure.
Yeah, it's right here. They got the whole history.
A crook. He's not such a crook. He was just. I think he was trying to feather his own nest at the expense.
Because the current guy is Kevin O'Connor.
Right, who's great, by the way.
But the first guy.
Kid's great.
Norm Abram was a spin-off, but what was the guy's name? They don't even have it here on this website because they want to...
Go to Wikipedia.
Yeah, I am on it because...
Oh, you are? Early days? No early days on that. We're going to get letters. I know a lot of other people out there know this guy. You see him every now and then on some advertisements for like drills and stuff like this.
All right, let me try another approach. Original.
Right.
This Old House host. Bob Vila.
Bob Vila, of course.
Gosh, this is such a wormhole, and it's actually a rat hole. I'm sorry, folks.
No, you know what?
You basically, you buy a house, so the schistosome worms come in. Sure. They go into the Peyer's, but they don't like it, so they remodel it. They get Bob Vila to remodel it.
Didn't taste good.
And they change the vasculature. So obviously there are worm proteins that are involved in doing this, and of course we don't know what they are. But can you imagine figuring this out? How informative it would be. Yeah, There's so much we could do, Dickson.
In my book, I hate to mention this, but in my book, parasites and plowshares. This plowshare concept comes up a lot. Here's a plowshare concept, because if you could alter the vasculature of the Pius patches favorably, right? Maybe you could increase the communications between the antigen-presenting cells and the immune system. Who knows? But
they also say that filarial nematodes also remodel.
That's right.
So, for example, proteins from Brugia malayi.
Right. Or Malayi.
Malayi. Please do correct me.
Coming from Malaysia, which is now called...
What?
I think it's Myanmar.
You're going to get emailed, Dickson, if you go on to these geopolitical things that you don't know anything about.
No, I think I do know this one. I think Malaysia has changed its name to Myanmar. Or was that Burma?
Malaysia is Malaysia. What's the capital of Malaysia?
Colombo.
Oh,
That's Sri Lanka. It's Sri Lanka. Sorry.
Myanmar is different.
You know what? You're going to edit this all out, aren't you? I hope. Otherwise, we'll be overwhelmed with geographers.
It used to be Burma.
That's right. Myanmar is Burma.
No, it's not Burma. It used to be, right?
Right. Okay.
So just so we didn't get email, I went and looked. Okay.
You know what? They're going to get email anyway, because they're going to, what is this guy doing on there? He's supposed to be a tropical medicine guy. Yeah. He doesn't even know what.
Anyway, Brugia malayi,
Malayi
also induces lymphangiogenesis. What's that, Dickson?
Well, angiogenesis is a general term for the increase in circulation of, and then you have to fill in that blank.
So you're making more blood vessels.
Yeah, well, you could do more capillaries, more precapillaries, more venules, more arterioles, more sinusoids, or in this case, more lymphatics. So Brugia malayi lives in the vessel lumen.
So that's good for this Brugia to make it to be angiogenic.
Apparently, it's making room for more.
And this is specific for the lymph nodes in these cases.
That's right.
Not
and
just
the lymph vessels. The lymph vessels.
And it says here possibly to transform these into a more suitable niche for the.
They don't know, but you know what? They live there a long time, Vincent. A long time. And the human equivalent, well, there are human examples of Brugia malayi also, but it's also used in animal models like Jerds, for instance. Okay, the human one that really worries everybody is Wuchereria bancrofti.
Which?
Named after Wucher and Bancroft, that I know.
That took me a while to get. Wuchereria bancrofti. Wuchereria bancrofti. Wuchereria, you from?
Yeah, Malaysia.
No, Burma. No, sorry, Myanmar.
That's right. That parasite can live for tens of years inside the same host.
Right. Now, the next one I knew would be your favorite.
Well, yeah.
It's modulation of tissue vascularity.
That's right.
But the first example they give is not your organism.
No, it's the one we have on our desk. It's the one
we have in a tank on the desk here.
On the tank. This is a museum specimen jar, please.
This is not a tank?
No, it's not a tank.
It's a glass tank full of what?
It's a museum jar.
What's in it?
There are the heart and lungs of a dog.
It's got this thread-like thing
and they've
in it.
got lots of little thread-like things in their heart.
What is it?
It's dirofilaria immitis.
Wow. And where did you get that?
Well, I inherited that from the original developer of the parasitic disease course at Columbia University's medical school. And
Harold Brown?
I believe this was an original specimen jar from either Dr. Harold W. Brown or Roger Williams, who also worked on a Vector-Borne. This is a Vector-Borne nematode infection, by the way. Mosquitoes carry this infection from dog to dog. Did you know that?
You probably mentioned it on the episode.
This is one of the few Vector-Borne.
Did we talk about dirofilaria?
Yeah, we did. We did.
Now this is a worm. What kind of worm? No, this is a roundworm.
It's a roundworm.
I can tell by looking at it.
That's right. So that makes it a
nematode.
Correct.
It says here that difilaria causes cardiopulmonary
insufficiency,
parasite residence in the arteries, and they secrete antigens that affect the vascular epithelial cells, vasodilating and decreasing permeability to monocytes. So they make the blood vessels bigger so they can sit in there, and they don't want the monocytes to come in.
And they exclude the immune system.
Yeah, so that we clear them.
Isn't that clever?
Brilliant.
Okay.
Dilation of blood vessels enables the host to accommodate additional worms, of course, right?
Right. And by the way, when dogs get this infection, they become short of breath, they become lethargic, they can often die from it.
Yeah.
And so...
Look here, Dickson. Look at how they're snaking through the blood vessels in your specimen.
Right. I know.
I'm going to put a picture of this. What?
Good.
What did you say?
I said I know this very well. We used to use this in our teaching.
I know, but I'm saying it so the listeners understand. I'm going to show a picture of this in the
sure.
episode.
I mean, you've got to do that.
This is a great specimen.
It is. It's one of the few I salvaged.
I'm going to bring my video camera in and record you explaining. You got this one. Plus, you have a lot of other specimens out there in your office.
Yeah, that's true. That's true.
Which make for a horrible smell.
Doesn't it? Unless you close them off like this one is. So, wait a minute, Vincent. We're not done yet with this parasite.
I'm not done.
No.
Okay, go ahead. Dogs get short of breath.
And dogs
kill them?
These are domestic dogs mostly, right?
Will it kill them?
So, it can.
Can you cure it?
Yes.
Drugs.
And in fact, your wife worked for the company that came up with the cure for this thing.
Ivermectin?
That's correct.
HeartGuard.
That's exactly right.
So you just routinely give HeartGuard to your
Before
dog?
that was dichlorvos.
You don't just check. You just give them HeartGuard all the time.
Well, if you do that all the time, you'll select for resistance. But
is there a diagnostic?
There is, yeah, because these worms produce offspring right in the blood supply. They're called microfilariae. And if you do a blood smear or a KNOT test, KNOTT test, you can find the microfilariae and make the diagnosis. But I think you could do an MRI on the dog and actually see the worms.
Pretty expensive.
Some pet owners don't care.
When I visited Virginia Tech
in
March, I think, they gave me a tour of the veterinary facility.
They have
vet
great
school there.
MRIs.
And they were doing an MRI on a cat.
Exactly.
It's this huge magnet, of course.
Exactly.
And it's in a room.
No, they were doing a CAT scan. I'm sorry.
That's right. The cat was lying there.
Right.
Sedated with things going in it.
Right.
And it was in the area. And then on the other side of the glass, no less than 6 people monitoring this whole thing.
That's right.
You can imagine how much that cost?
I can't.
But it's fluffy.
It's fluffy, and fluffy is important. Some people leave their whole fortunes to their pets when they die. You know that, don't you?
Well, how can the pets?
I have no idea.
Someone takes care of it.
So maybe we want to put this on the show notes also. There was a How It's Made show very recently.
How It's Made?
How It's Made.
Oh, I like that. That's a good show.
I love that show. You know what they did?
What?
They built an MRI for us.
Oh.
And you know what? You know how they cool it? To make the magnet superconducting.
I think they use liquid nitrogen, right?
Oh, no. That's not cold enough, Vincent.
No?
What's the coldest substance we know of?
It's a scorned person.
You said liquid hydrogen?
Is it liquid? No, not liquid
What's
hydrogen.
the next element up? Hydrogen.
Wait.
Starts with
lithium?
Hydrogen.
Helium.
Helium.
I'm trying to sing the song, but
that's right. No, it's the periodic table, right?
Yeah.
So hydrogen is, if you put it in liquid form, it gets down to about, I think it's 100 and... Let's look it up. What is the liquid hydrogen's temperature? Because someday we'll have cars that run on liquid hydrogen, right? Because we now have hydrogen-powered cars, and they'll just keep getting more sophisticated with it, and pretty soon you'll be able to compact hydrogen down.
What do you want, centigrade, Fahrenheit, or K?
Well, give me a K.
Well, it's 20 K, which is minus 423 Fahrenheit.
Minus how many?
Minus 400 Fahrenheit and minus 250 Celsius.
So what is liquid helium?
That's hydrogen. Okay.
What's helium?
Helium?
Yeah.
Liquid helium, on the other hand,
right.
4 Kelvin.
Exactly.
4 Kelvin or minus 452 Fahrenheit.
That's the coldest liquid gas we know of.
Is that right?
Yeah.
Are you sure?
We are.
That no one's going to send you
No, it's
email?
true. No, I know someone who actually is the... he sells the cryogenic apparatus that's necessary for the MRIs to function.
Do you know why it... So basically it needs that temperature to become liquid. Do you know why?
Because they want to convert the magnet to a superconductor.
No. Why does it have to be so cold to make helium liquid? Because of the attraction between the helium atoms.
Oh, sure.
They're weak.
Well, on the show, How You Make an MRI Machine, they showed it filling up with liquid
helium, really?
Liquid helium.
So how do you chill it to become
Well, liquid helium is not that common, by the way.
You buy it, I presume.
Yeah, helium is not that common.
But so I think the trucks come and deliver it already liquefied.
They do.
So they must be chilled somewhere in a plant, right?
That's right. That's exactly right. So what is liquid nitrogen, by the way, just for argument's sake? I know it's about 170.
Is that what I had suggested? Liquid nitrogen?
No, we have liquid hydrogen, liquid helium, and liquid nitrogen.
No, that was my first answer, right? Liquid nitrogen?
Right.
Liquid nitrogen.
Not so cold, actually.
What's the Kelvin? Because it's only 77 Kelvin, which is higher than helium or hydrogen.
Exactly. So it looks like, well, what does it look like? Is there a trend here? Can we say something about if you liquefy a gas, what is liquid CO2?
You mean dry ice?
Yeah. Well, that's not dry ice. That's solid.
Is there a liquid CO2?
Let's see. Or maybe there's not. Maybe it sublimes into a gas from a solid. Is that true?
Carbon dioxide. Yeah, because we have it right outside the door here. I could go get you some.
You can't liquefy CO2. I think it's either a solid or a gas. This shows you the extent of our chemical knowledge here, folks. I hope you're all having a good laugh, because I know I am.
So you want to know
can you liquefy CO2?
Okay.
I don't think so.
There's actually a thing here, how to liquefy CO2.
Really? Okay, well, I stand corrected. There's another example of a gas.
Well,
right?
I'm not sure it says you can make it liquefy. Why do we buy...
solid is even more compressed. Handle. It's even more compressed and easier to handle. Absolutely easier to handle. Of course it is. But it doesn't liquefy as it melts, does it? It sublimes off into a gas.
Actually, if you put it under high pressure, you can liquefy it. Okay. 56 atmospheres and 20 degrees centigrade. Yeah.
Right. And what is its temperature that you achieve with that?
It's 20 degrees centigrade.
Oh, I see. Okay.
You just pressurize it and it becomes liquid.
Not very cold then. 20 degrees centigrade. You can put your hand on it.
Room temperature.
You can put your hand on it.
68 degrees.
Right.
Fahrenheit.
Liquid CO2 is at room temperature. Isn't that interesting?
Well, it's at high pressure. 56 atmospheres is a lot of pressure.
Put your hand in there.
Well, no, you can't because it's under pressure. You can't put your
Let's go back now to the concept of using an MRI to discover the presence or absence
started 10 minutes ago.
Dirophilaria emittus in a dog.
And I said, this is really expensive.
Well, what about a
That's why our
test?
dog, every time it goes for it, they just give it HeartGuard.
I know, but you shouldn't treat something if it's not there.
I totally agree with you.
Too many physicians.
Listen, there are definitely veterinarians listening,
and they know better too. They said, "Well, we'll do a NOT test on your pet, KNOTT test."
But would you do that routinely? Because if you don't want to wait till they have symptoms.
What about immunological tests? I bet you we could look up right now as to whether there's an immunological test for Dirophilaria on the market. I don't know the answer because I'm not a vet, but it would be interesting to say whether there is or not, because here we are trying to diagnose this infection, right?
Mm-hmm.
We're drifting a little bit off the theme of the paper, but it's worth going astray a little bit.
It's called a NOT test.
Oh, I know that test. It's not an immunological test.
It's not?
No, it is not. It looks for the microfilariae.
Yeah, that's right.
So is there an immunological test?
Yeah, there's an antigen test.
There's an antigen test.
And most practices have supplanted the NOT test.
Okay. So that means...
It's an ELISA-based test.
It's an ELISA-based test. So that's what you do on your pet first.
You detect antigen.
Right. So that just tells you if it's been exposed to the infection at one point or not.
Well, if you have antigen, not antibody, Dickson, antigen.
It's an antigen capture test.
Yes.
Okay. Antigen capture test.
So we're looking. So if you have antigen, you probably have active
absolutely fine.
infection.
Yep.
Okay.
100% agreement.
Anyway, the next organism in this category of modulation of tissue vascularity is a something
obscure
called T. spiralis.
Right.
What does this do?
Right. Well, not as the adult stage in this case. These are the adult Dirophilaria doing that.
Right.
The larva that lives in the muscle tissue, and we've reviewed this extensively in our treatise on the Trichinella organism and how it makes its livelihood. This worm creates a home for itself in striated skeletal muscle and modulates the expression of many genes as the result, and it develops into something called the nerve cell. And the nerve cell...
A myocyte, right?
Yeah, well, it's not just a myocyte, it's a myotube. It's a myotube, and a myotube is a collection of former myocytes which fused together to make the myotube, okay?
And we talked about this extensively.
Yes, we did. And we can do it again, too.
Because this was your shtick.
To to the point of ad nauseum. Okay, but the point is that in this review, here's another example of a worm controlling angiogenesis. But what vessel is being angiogenicized?
I knew you were going to get into that.
And of course, that's what we worked on that one for quite a while before we determined that what the vessel was not a capillary, and it was not arterial, it was not a venule, it was not a vein or an artery, it was a sinusoid.
Really?
Loose junctions. Now you're going to go back to that concept of loose junctions, right? It has to be able to exchange with its host the waste products that it creates, and it has to be able to get nutrients. And this is how it does it.
It says here that
so we think.
During early stages, you can see higher levels of VEGF, vascular endothelial growth factor.
That's correct.
Is that induced by Trichinella?
Oh, yeah.
Do we know how?
We don't. We think that the worm communicates with the nucleus of the host in some way, though. Inside this developing...
You should write a grant to work on this. You're retired, right?
Well, yes. I wrote a grant on that, by the way.
You ever get the itch to go back in the lab?
I do, yeah, every now and then.
Then you scratch it and it goes away, right?
Well, I miss the action of discovery.
And the thrill of defeat.
Well, I knew that one very well. You don't win every time you ask a question. You have to learn how to ask the right questions, and if you keep them simple enough, you can get those answers, right?
So somehow the worm induces...
Right.
Production of VEGF. We don't know how.
Which VEGF? Now, it turns out
there are more than one.
There's VEG-A, VEG-B, VEG-C, VEG-D, VEG-E.
And cancers do this also, right? They induce VEGF because they want their tumor to be vascularized.
That's correct, but which one do they induce too? So,
is the mechanism similar?
The mechanism
is not known for Trichinella.
It's not similar. It can't be similar. Tell me why.
Because we don't make a tumor with
because
Trichinella.
Tumors don't elicit sinusoids; they elicit capillaries.
Okay,
that's a different VEGF. VEGF-A, VEGF-B, VEGF-C.
Does anyone work on Trichinella angiogenesis anymore?
I don't know. I hope so.
I'm going to look it up while you talk. Go ahead.
I don't know of anybody with a grant that's now working on it. I did. They asked me once. The Wellcome Trust asked me once to be an external reviewer on a grant that was about the subject, and I thought it was an excellent proposal, but it didn't get funded. It fell just below the funding level. So,
wow.
Dickson,
I searched for Trichinella and angiogenesis.
Yeah.
I got 5 papers.
Right.
And the most recent one is 2013.
Oh, good.
Thymosin dependent and
independent
angiogenic.
That's a Chinese group that's
induction
of
Trichinella and their cell formation.
Yeah, right. I reviewed one of those papers for publication as well. So,
and there was one in 1991,
right,
with your name,
that's
you and
right.
Baruch.
That's right, Alice Baruch. That's right. She was the one that did the plastic casts that proved that it was sinusoids and not capillaries.
Vascular casts. Okay.
Right. So VEG-A...
Not much activity, in other words.
Right. VEGF-A induces cells that have... I'm either going to say it has tight or loose junctions. It's one of those two things. And another VEG something, B or C or D or E, induces cells that have loose junctions. Okay? So we could look it up. I mean, I just gave a talk on it, but the... I
think you did, didn't you?
The data on that
at USUHS.
That's right. That's right.
Which is Uniform Services Health Sciences University.
That's right. I reviewed the literature on that. So I have to pull up the slide in order to refresh my memory as to which VEGF induces the tight junctions. But we know which one does this. And so if it's one that doesn't do that, one which induces loose junctions rather than tight junctions, that's probably going to be the one that Trichinella affects the most in the host's genome. So here's a parasite nematode controlling host genomic expression of VEGF to suit its own needs. That's remarkable biology.
This is the theme.
But there's another one that wasn't mentioned.
Which is
that? That was Onchocerca volvulus.
Mm-hmm. You're right. What do they do?
Then we have to go back to the adult stage for this one. Now that's another filarial parasite. Okay, it's related distantly to Dirofilaria.
Right.
Okay, so the adult of Dirofilaria induces dilation of vessels and modulation of the endothelial cells, and the adults of Onchocerca volvulus, they live in nodules under the skin, and they're completely vascularized. And no one has actually done the same kind of work that we did with Trichinella on Onchocerca nodules to see the extent of the vascularization, when it starts, what kind of vessels are present. But these worms are nematodes. They don't eat the tissue of the host. They absorb nutrients through their cuticular surfaces is the way we think they live. And they need a lot of circulation surrounding them to bring a plethora of nutrients to the parasite as it sits under the skin. So again, we're missing the whole biological story on this parasite because, well, we know the reason why, because the funding streams are not favoring this kind of research any longer. But it's too bad, because if we knew, then we might be able to get rid of Onchocerca once and for all by interfering with its angiogenic responses, rather than having to give ivermectin all the time.
Right, the last one.
The last one.
Tuning of granulatomas responses.
Or granulomas.
Granulomatous.
Granulomatous responses.
What's a granuloma, Dickson?
A granuloma is a collection of white cells around an offending thing. Now, the offending thing can be anywhere from a toxin to a virus, to a bacteria, to a fungus, to a protozoan, or a helminth.
These are typically chronic infections.
They are.
Right?
It takes time for these to develop, right?
Tuberculosis, you get granulomas in your lung.
You absolutely
They may calcify, and then it's
and
called the Gone complex, I think.
So there's there's a guy down at...
Robin will tell us.
Well, that's true. Down at NIH, his name is Alan Scher, and Alan began his career working on schistosome.
I know Alan Scher. I met him through my wife.
No, he's a great guy. And he began his career working on granulomatous responses that the eggs of schistosomes induce in the tissues. And now guess what he works on?
What?
Tuberculosis.
It says here that the helminths actually prefer to have granulomas.
Oh, yes.
Because if you inhibit them experimentally in animals,
it would be even worse.
Egg, for example, S. mansoni egg excretion is reduced.
Right.
Without a granuloma.
Oh, because they require TNF, tumor necrosis factor. They require it as a growth factor for their own biology. So they have hooked their own biology into the host's biology of immune responses. That's the next level of spy versus spy. That's a remarkable adaptation, by the way.
So they're saying here that schistosomes and also Taenia, they downregulate the immune responses that do not lead to granuloma formation and favor the ones that lead to granulomatous responses. And this is done by parasite proteins that alter host mRNA patterns.
Sure, and chemoattracting agents that they...
So they don't want inflammatory cytokines produced. So they inhibit those, but then they want the type of the immune response that allows the granuloma to form. They like granulomas.
So here's the deal, though. These are long-term infections, okay? Schistosomes are long-term. They can last up to 20 years. So let's look at a new infection, a middle-age infection, and an old infection.
Mm-hmm.
And let's look at the size of the granulomas. All right? Well, these are Th2 responses.
That's correct, right.
But
Th2 means Th2 is a type of T cell.
Correct.
That produces certain kinds of cytokines.
Correct.
That would favor,
cell-to-cell killing or antibodies?
Antibody production is a type 2.
Okay.
Yeah.
So there are antibodies produced during this infection around the granulomas, but there are also type 1 responses. So it's a mixed cellular response.
Yeah, the worms don't like type 1. They don't like type 1.
They don't. But the eggs of this might, because in the egg, in the growth pattern of the parasite, there are some of those growth factors that are produced by type 1 responses are required for egg production. All right? And Alan Scher worked on that a lot and was able to pin down some of these response patterns. All right? So what I'm trying to say is, though, for the early infections, if you looked at the size of the granuloma, you can measure the volume of these granuloma. And you can also measure the kinds of cells there as well by using cell markers of various sorts. They were huge. Mm-hmm. And it damages a lot of tissue around the granulomatous response, so that innocent bystander effect of schistosomes in the liver means that the parenchymal cells are affected as well. In a middle-aged infection, let's say 10 years down the road now, and you look at the size of the granulomas that are produced by the eggs that were just shed by the adults, versus the granulomas in the beginning of the infection that were shed by the eggs, that were induced by the eggs shed by the adults. It's almost half the diameter, almost half the volume. All right? And then finally, long-term infection, way out, maybe 15 or 20 years down the road, these granulomas are absolutely small and almost imperceptible around these eggs. So there was a general feeling among the immunologists in the day, as I would say, of an inhibitory set of T cells, which were inhibitory towards the formation or the size of the granulomas. All right. And there was a whole science built up around that concept of inhibitory as well as stimulatory T cells that would favor the formation of a granuloma or favor the dissolution or the non-formation of a granuloma. And I'm not sure how immunology has progressed since then to say, well, that's probably not the case anymore because we know that certain T cell populations can modulate their own sizes as time goes on. In a host that's 30 years down the road and infected with schistosomes, there's very little innocent bystander damage to their parenchymal cells. And so the parasite is almost coming into modulatory peace with its host. But by that time, of course, the tissues of the host are compromised in all kinds of other places. Eggs don't just stay in the liver, they go into other places throughout the body as well. And it's really too bad because when that happens, by the way, you get compromising of the ability of the liver to detoxify all those bad products that are coming back from the gut tract, from the microbiome, and the patients start to develop toxic brain syndrome, for instance, where the toxins actually reach the brain tissue and then start to affect the functioning of neurons. So big, complicated disease. Schistosomes are really big and complicated, and just raises all of those issues by saying, if we knew more about this, we could do that. If we knew more about that, we could do this. Knowing more is the essence of being better prepared to deal with parasitic infections and understanding the details of their lives at levels that we currently don't have, but we need in order to complete these pictures. So that's a great paper that you selected. Vincent selected this one, by the way. I did not, and I'm happy that you saw this as an opportunity.
Good jumping point for discussion.
It's fantastic. Yeah,
and it's easy enough to read for any of our listeners.
You got it.
It's a nice summary, so check it out.
Pearls.
Do some email?
Absolutely. As long as it's compliment. No, that's
Colin writes, hi, is there a parasite known to kill all virus it encounters?
A virus?
Yes. No, I don't know of a parasite that kills a virus, because a virus is a parasite itself. Then he asks, is there a virus known to kill all parasites? So there are some known viruses of parasites, and we've discussed them on TWiP before.
Yeah, but they're double-stranded RNA viruses.
Well, so what?
That's exactly so.
Come closer to the mic. You need to stay.
Well, I have to be able to read my iPad.
That's not my problem. You need to stay closer to the mic.
I'm working on it.
So there are viruses of many parasites. Not all have been discovered, but the viruses of
do they give advantages?
Giardia, for example, remember?
Or Trichomonas, remember?
Trichomonas.
Sure. It had something to do with inflammation.
Yeah, that's right.
I think if you get rid of the virus infection, you get rid of the ability of Trichomonas to make an inflammation.
Yeah, so I don't think these necessarily harm the parasite, the eukaryotic parasite.
No, they're probably essential for carrying out some of their life cycle stages.
Yeah.
That's right.
Has any virus been found to have parasites yet? And if so, do they have virus? And so on and so on.
Well, that's a Mimi virus, right?
So there are viruses that co-infect cells infected with these giant DNA viruses like Mimi viruses.
Right.
They interfere with their replication. They're also dependent on them. Right. So they're satellites of a sort. Sputnik is the name for one. They only can replicate in cells infected with the bigger DNA virus because the bigger virus provides functions needed that are missing in the smaller virus genome.
Right.
And they do interfere with the replication of the helper virus.
Huh.
And they're thought to be in part important for modulating communities and so forth.
Right.
But then that virus, that Sputnik type virus doesn't have any viruses that depend on it, as far as we know.
Yeah.
So it doesn't keep going on and on.
Right.
I know these questions may seem a little perhaps different, but what do you guys think? So I don't think it's gone very much beyond the parasite and virus that infects it.
Right.
You understand what I'm saying, Dickson?
In general, yes, I do.
You want to read James's? It's a very long one.
All right, James writes.
Read clearly, speak friend, and enter. Stay near the mic.
I'm going to try my very best.
Maybe we need to set you up with a stand, huh?
Maybe
we need to print it out for you.
No,
Go ahead, read
James writes. I'm not a veterinarian or a fish farmer, but I trained, he meant to say, as a marine scientist, including a large part of my studies at the Institute for Aquaculture, and worked on the production of a vaccine. I don't know what the Institute of Aquaculture is, but we're going to find out, I think. A bit of feedback concerning how TWiP 70, which was great, as always. Yeah.
A bit of feedback concerning TWiP 70, not how.
Okay, fine. I'll read that again. You can edit it out. A bit of feedback concerning TWiP 70, parens, which was great, as always.
I'm not going to edit it out. It's too much work.
Oh, you're going to make me look bad again. Okay, fine.
That blew that out.
Right. Apologies, if anything, has be discussed across TWiCs.
Has been.
No, it says has be.
Yeah,
sorry,
no,
I'm reading it exactly as you criticized me before.
Can't you
screwing up?
Improvise?
I try. Let's try it again. Apologies if anything has been discussed across TWiCs. Feel free to center on interesting bits, parens, if any. Zoonotic or non-zoonotic invasive bodies. Just as significant, if not greater risk of invasive aquatic species likely to be through ballast water and shipping. This has already had significant impact across the Atlantic. Carcinus manias, the green crab, European crab, has been well studied and decimated your soft-shell crabs in parts of America. They hitchhiked as larvae in the ballast, which, when expelled at shore to raise the height of the ship to enter, so do the larvae. Conversely, America then sends signal crayfish over by the same means to pay Europe back. No, we didn't do that for that reason. But by the way, we also got zebra mussels that way too. This is why in Australia, in addition to strict animal import rules, i.e. never almost, ships are ordered to remove ballast much further out at sea in order to protect the barrier reef, as well as severe consequences for shipping firms. Ship captains flaunting this will have severe consequences. Non-nationals face banning from the country and its territorial waters, a career ender.
Can I ask a question?
You may.
What's the function of ballast? To keep the ship low?
Yeah.
To keep it balanced?
Yeah.
So if you didn't, it would be high up and could be knocked over in a storm, right?
Exactly.
But when you come into a port, that's not an issue because...
It's not an issue because you're going slow enough and stuff like this.
Okay.
But if you dump that bilge water someplace sensitive,
of course.
Like, for instance, in Lake St. Clair, which happened, by the way, and that's how the zebra mussels gained entrance into the United States, then you've got some problems because it's now everywhere. Aquaculture versus rearing. Unlike true eels, the Monopterus albus does not require migration to, for example, in European species, the Sargasso Sea, in order to complete its life cycle. However, I suspect, although I cannot find any exact confirmation, that M. albus has still yet to have its full life cycle closed in aquaculture. Hence the phrase farm-raised or reared being used. In my quick search, the only specific confirmation to captive either way to breeding of M. albus was a negative one, and he gives a website, with a few U.S. government agencies referring to escapes from aquaculture farms. But these could refer to those that escaped from a farm of another species, for example, fish. Sorry, I can't be more specific. Most of my documentation is in storage in Scotland. Hello again, doctors. In Anguilla, it actually still requires the capture of live elvers, young, and ongrowing in capture. They cannot be bred. Despite this, is similar to the maturation of salmon, which can have its life cycle closed. The closing of Anguilla species' life cycle has still eluded scientists. I still find one reference to the use of hormone treatments for M. albus, but I am skeptical, barring further documentation. Okay, now fresh versus salt. This is a long email. DD, brisked over there being fresh and saltwater eels. Anguilla, European eels, that are part of the true eels, Anguilla, and are normally fresh living but migrate to sea for spawning. European eels migrate all the way to the Sargasso Sea. Satellite tracking of eels. And it gives a website.
Oh, cool.
Larvae return via the Gulf Stream.
You can track satellite eels by satellite?
Yeah, there you go. You have little...
Nice.
The same is true for the American eel, Anguilla rostrata, another website given, which also travels to sea for spawning, although the journey is less dramatic than its European cousin. The whole of the Anguilla is often described as freshwater due to the greatest part of its life cycle being freshwater. Eels have strict protection in the UK. It means criminals try to harvest elvins at night, leading to police waiting in the dark for them. Polyculture. You mentioned polyculture. It has been known on developing areas for pigs to be cultured in wooden sheds. On the lower level, chickens are cultured. Both floors have gaps between floorboards for the excrement to fall through to the lower level. Below the fish, it is built over rivers, so below the chickens are small fish cages. I see. It's like an ecosystem based on feces. This also kind of side arches into DD's new podcast. First episode was great, but I will save that for a separate email. Right. Didn't catch many eels, 100. I can beat that. That's another title for his editions. There was a now infamous article of contamination in fish feed and effects on human health due to PCBs, that's polychlorinated biphenyls. It rose to stardom due to the BBC being reprimanded for its use as part of a documentary that was highly biased. It involved a sample of less than 10 fish, 5, I think. It was a farmed versus wild comparison, assuming feed would result in changes on the chemicals composition of the fish, basically, is what I think he meant to say. Of the fish sampled, some were from America being reviewed as part of EU policy. Wait a minute, I think I skipped a sentence on this one.
Yeah, you did.
Of the fish sampled, some were from America, some UK. The one farmed fish that was ordered, it turned out, wasn't even farmed, never specified when ordered. It got published and was at the institute supplying the fish.
No, you skipped 2 lines.
It's this densely written.
I
know. I'll go back. It got published and was at the institute supplying the fish.
No, it was being reviewed.
I'm sorry, I keep skipping that line. Being reviewed as part of EU policy. The blunder only got found out through coincidence of a scientist being on the EU review board, also being at the institute supplying the fish, knowing they only used wild samples of that species and later confirmed this. Great to hear all of your collective podcast, James. Too warm at about 18 degrees centigrade. Whew, that was a long one.
18 is not that warm.
Not really,
Maybe
but
for
that
him
depends
it
on what
is.
he's used to, right?
Yeah. It was cool.
Yeah.
Lots of good stuff there. 100 eels, huh? Did you catch 100 eels? Did you say that? No?
In the Delaware River, where I do a lot of fishing, especially the upper reaches where the eels start to gather, there are eel weirs. And
what's a weir?
A weir is a collection of, in this case, it's a collection of stones that have been piled up in a V-like pattern downstream, so that the eels as they migrate towards this eel weir get sort of herded along into a narrower and narrower and narrower zone until finally there's a wooden platform.
Yeah. And
the wooden platform at the bottom of it has a basket. And the eels actually migrate up out of the water across the platform. Yep. And into the basket. And every now and then the eel fishermen come along and harvest. Then they take them back to these little feeder streams of the Delaware that are dammed up. And they're quite deep, actually, some of them, maybe 20 or 30 feet deep. And they keep putting the eels in these holding pens until the price of eels actually go up. And then they quickly harvest and bring them to market.
Seems a bit...
It's... You're going to say draconian?
No, it seems a bit unfair, no?
It's unfair if you're an eel, that's for sure. Yeah, that's very true.
All right. The last email for today is from Rachel, who writes, "Hello, TWiP team. I would like you to know that there are veterinarians listening to This Week in Parasitism."
Good.
By the way, we just read one of Rachel's at the beginning.
We did. So she's a double hit for Rachel.
This is the first one, so it just tells us about her.
Okay,
I am a veterinarian, immunologist, and steady Twix listener. I have recommended all 3 Twix podcasts to many colleagues, veterinary and otherwise, over the last year since I came across them for the first time. I find TWiP in particular is interesting to me, due in no small part to my veterinary training.
Oh, nice.
We spend a lot of time on eukaryotic parasites, especially those with zoonotic potential.
Absolutely.
Additional episode 70 notes, I would really like it if you would spend more time explicitly on the non-human side of zoonoses, as you brush up against it during many episodes, but rarely spend much time on it. Also, add me, along with letter writer Heather, to the list of people who would love an episode here and there devoted to aquatic or aquaculture themes. My background is largely in the immunology and diseases of fish.
Nice.
Keep up the good work. I like having Twix in my rotation of cell culture listening.
Terrific.
Well, we really appreciate your telling other people about it. That's great.
Yeah.
And Dixon, I've told Dixon many times we need to do non-human parasites and water-based.
Yeah.
And if you can't do it, Dixon, we'll need to get somebody who can. Right?
Well, I'm not trained in veterinary parasitology, that's for sure.
Well, you can certainly talk about it because we have a paper. We could talk about it.
Of course, I can talk about it. I mean, you can make the bridge between viruses that infect animals and viruses that infect people. So why can't we do the same for eukaryotes? And we just did, actually.
You're a scientist. You're
the Dirofilaria immitis is a zoonotic infection.
Where does it come from? You said it was mosquito transmitted?
It's mosquito transmitted from dog to dog. They pick up the microfilaria.
Are they the reservoir host?
They're not a reservoir host. They are the host.
Who's the reservoir host?
There aren't any.
So they are both the host and the reservoir?
No. You have to have another species of animal to have a reservoir host.
Well, if there is no other species, you're both then.
No,
by definition,
then there is no reservoir host.
Fine. So be picky.
I have to be. In this sense, I really would like to be. So,
all right.
But we are an aberrant host because when we are bitten by a mosquito that has the infectious larvae in the proboscis, right?
Yeah.
And she deposits them on our skin and then withdraws her proboscis, and the larva crawls in and infects us. Mm-hmm. Typically, these worms travel to our lungs and they get stuck. They can't go any further. And they realize, also, much to their chagrin, that we're the wrong host. We are the wrong species. And so they curl up in these little round configurations. And so if you went online to Google and typed out lung coin lesions, they can often be mistaken for cancers. Because when they die, they calcify. Okay, and you get these little circles of calcification. Now, what the hell is it? And it turns out to be a dead Dirofilaria immitis in a lot of cases. Mm-hmm. And nothing needs to be done if you could say for sure that's what it was. But, of course, you have to biopsy it to find out.
It says here a lung coin lesion is a mass in the lung smaller than 3 centimeters in diameter.
Right.
Can be an incidental finding in about 0.2% of chest x-rays.
Yeah.
Most commonly represents a benign tumor such as a granuloma or hamartoma. 20% of the cases, it's a tumor.
Yeah. But in the rest of those cases that go undiagnosed, it could be a dead, calcified Dirofilaria immitis developing adult that didn't succeed.
In a dog, you mean?
No, in the humans.
In a human?
In humans.
We get this? We are infected with Dirofilaria?
We can become infected with it, but it is an aberrant infection. It doesn't often result in a lung or a heart infection.
It's a dead end.
It's a dead end.
Okay. Dixon, where can you find TWiP?
All over.
All right. You can find TWiP on iTunes and also at microbeworld.org/TWiP.
That's true.
Why don't you know where our podcast is?
Because you're reading all of this. You know, if I could read it, I would.
I'm not reading it. I'm staring at the ceiling.
You can find it on iTunes.
So many times I don't have to read it.
You can find microbeworld.org/TWiP. Or you can just go online and type out TWiP.
Let me try that. TWiP.
It's easy to find.
Let's see what I get. The first tick is This Week in Photo.
Go on.
And the second is This Week in Parasitism.
A second.
And the third, we're not bad. The third one is a Wikipedia entry which says a TWiP is the 20th of a point. A typographical measure.
A 20th of a point?
I didn't know
Is that
that.
what we are? We're a TWiP?
Yeah, interesting. Anyway, yes, you can find them there or on Google, and we'd love to get your questions and comments. Send them to TWiP.
Would. Yes, we would.
TWiP@twiv.tv.
Bingo.
You can find Dickson Despommier at trichinella.org, medicalecology.com, is it? Or org? .org.
That's an org.
And verticalfarm.com, and also at urbanag.ws.
That's right.
Thank you, Dickson.
You're welcome. It was a pleasure.
I'm Vincent Racaniello. You can find me at my website, which is virology.ws. I'm also on Twitter, PROFVRR. Dickson's on Twitter, but he doesn't actually...
I don't twit.
Tweet? Who is tweeting for you?
I don't.
Someone is, because there's a...
I don't do it. I just don't do it.
All right. Anyway, you can get me on Twitter, PROFVRR.
I'm old school.
The music you hear at the beginning
school.
and end of TWiP is composed and performed by Ronald Jenkins. You can find his work at ronaldjenkins.com.
Excellent.
You've been listening to This Week in Parasitism. Thanks for joining us. We'll be back soon. Another TWiP is parasitic.